Hello, NEET aspirants. How are you all? Hope you are doing good. Now, it's a very important program we have started for you, that is NEET Crash Course program, in which you'll be covering each and every topic within minimum time. So, let's start our journey with the first chapter in zoology, that is Animal Kingdom. And be focused because we'll be covering each and every point of NCERT. So, let's start our journey with first lecture, that is Animal Kingdom. Now comes a question. How are you going to classify these organisms? What is the basis on which you can classify? Now, if a question comes to you, how many species of animals exist on Earth? Is it 1.7 million or 1.8 million, or is it 1.2 million or 1.5 million? So, most of the students will be thinking it is 1.7 or 1.8 million, but it's not correct. It is 1.2 million species of animals. If we take all organisms, that is around 1.7 to 1.8 millions all organisms. But if we talk about only about animals, it's 1.2 million species of animals. So now such a huge number of species, not numbers of the individuals, it's number of species I'm talking about. So such a huge number of species has to be classified. So for that you should have some basic parameters to classify the organisms. What are those general features on which you can classify those organisms? Those few are the level of organization, body plan, symmetry, segmentation, germ layers, and coelom, and the notochord. Now let's see, there is kingdom now. So now we belong to what? Kingdom Animalia. We all belong to animal kingdom. So let's see, animal kingdom, if I talk about, all animals are multicellular. All animals are multicellular. Very important point here. So now when all these animals are multicellular, they have to be classified into some criteria. For that we have level of organization. Level of organization comes as cellular level and tissue level, or oblique organ system level. Here you see the symmetry. Now on basis of two types of symmetry we can majorly classify. One is radial, other is biradial. Radial, if we talk about, it's coelentrates and ctenophores. Because if we talk about asymmetrical, then it is Porifera. Bilateral will be the rest ones, including the Platyhelminthes, Aschelminthes, Annelids, Arthropods, Molluscans, Echinoderms, Hemichordates, and Chordates. Even Echinoderms also belong to Bilateria group, because in the larval stages they had the bilateral symmetry. Okay, so let's proceed further and study each and every point in detail. So level of organization, if I talk about the cellular level is found in Porifera. Cellular level of organization is found in Porifera here. So what are Porifera? Porifera are the sponges. It is found in Porifera, that is sponges. Now let's see why we say it as a cellular level. Because these organisms have only cells. They never form tissues. They never organize to form tissues. So they have only cells which will perform each and every function of the body. And that functions may be to carry the nutrients, storing the food. Okay, and maybe some cells are involved in nutrition also. So we'll talk each and every one in detail in Porifera. So let's understand first. This is the cellular level, means each and every function is performed by cells. They never organize to form tissues. Tissue level is found in Cnidarians and Ctenophores. Ctenophora and Cnidaria phylum will have the tissue level. That means now the cells have organized to form tissues. But those tissues have not further formed organs or organ systems. All functions of body are going to be performed by some specialized tissues. Like if I talk about muscular tissue, so they have some epitheliomuscular cells which perform some contraction in hydraulic organisms. So that means only tissue level is present. The tissues are coordinating with each other. Got it? So example of the phylum is Cnidaria and Ctenophora. Organ system or organ type. See, in Platyhelminthes you may think it is it's only organ system level of organization. No, it is both organ also and organ system also. Why? because organs have already developed in platyhelminths, but some organ systems like reproductive system and the excretory system are very well developed. So that means in that case you have both type of level of organization in platyhelminthes. So please write down organ or organ system level is found in platyhelminthes. Now what I'm talking about, let's see. If I say organ system level of organization is found in you are a human being, you have organ system level of organization. Yes. Now the question comes, don't you have organ level also? Don't you have tissue level also? And don't you have cellular level also? Now this is something which is the maximum in a hierarchy. You have the best being classified as having the organ system level of organization, but still you have organ level, you have tissue level, and you have cells also, but they have organized to form a complete organ system. So that is why you'll be considered as having organ system level of organization. Hope you understood this clearly. Examples now. Examples, if I talk about, it's Aschelminthes till chordates. So Aschelminthes till chordates, all organisms, all animals will have the organ system level of organization, including human beings. Hope you understood this very well. So let's proceed further with the next basis of classification, that is the body plan. Now suppose if question comes to you, what type of body plan you have? So simply answer is you have tube within tube body plan. Why so? Because if if you see this fish, it has mouth here and this has anus. So two main openings are present in the body. One is mouth, other is anus. So in that case it has what? Tube within tube body plan. But if the question comes, which type of body plan is present in sponges? So cellular grade or cell aggregate body plan will be present in the porifers. Porifers will have this type of plan. Why? Because the body itself is composed of only and only cells. The cells are having some division of labor. Cells have some division of labor. So that means they are coordinating between themselves. Okay. For example, some cells are specialized for storage purpose like thesiocytes. Some are there which are helpful in nutrition, that is trophocytes. Some cells are very much specialized for moving the water in that spongocoel and through the inside channels. They are called as collar cells or choanocytes. So in that case, you see many cells are individually doing something, but they are coordinating at least with themselves. So that is called division of labor in them. So they have some division of labor. Next comes the blind sac body plan. So you see blind sac body plan. What is that? Blind sac body plan means only one opening is there. Only one opening to the exterior. Only one opening to exterior that acts as mouth and anus. That acts as mouth oblique anus. Very important question. Here you see it's only one opening. The only one opening is acting as what? The mouth also and the anus also. Ingestion also takes place from that. Even the waste also has to be thrown from that opening only. So it has blind sac body plan. Now a question comes, in which phylum you will find blind sac body plan? So examples are Cnidarians, Ctenophores and Platyhelminthes. Please write down Platyhelminthes because many students do forget about Platyhelminthes. So Platyhelminthes also have blind sac body plan. Please remember. Next comes the tube within tube body plan. So tube within tube body plan is present in all organisms starting from Aschelminthes onwards. So let's talk about tube within tube body plan. So what is this? In this case, the separate opening you can see. There is a separate opening. And the separate opening is of what? This is the mouth here, and this is the anus here. So you can write here mouth. This is one opening here, and this has anus. So two separate openings are present in tube within tube body plan. Two separate openings. Two separate openings in tube within tube body plan. Now this tube within tube body plan is present in examples. Which phylum you will find? It is present from Aschelminthes till chordates. Till chordates, you all have tube within tube body plan. Hope you understood this point. So tube within tube body plan is present in Aschelminthes onwards till chordates. All have tube within tube body plan. That means they have separate opening for mouth and anus. So this was all about the body plan. Even further also you can study tube within tube body plan. Those organisms which have tube within tube body plan, they are further classified into protostomia and deuterostomia. But that's not in your course. So let's just have a glimpse of that. Protostomia means in which the mouth develops first from the blastopore region. The blastopore region will be present in the, you will study the gastrula stage in the reproductive system, that is embryonic development. In that case, you will see the morula, then blastula and gastrula stage. In gastrula stage, you will see there is some structure called archenteron, that is primitive gut developing. So the opening of that primitive gut is called as blastopore. So if the mouth forms first from blastopore, it is protostomia. But if the anus forms first from blastopore, that is called deuterostomia. So we humans are what? Write in a chat box. We are deuterostomes or protostomes. Write it. So let's see now. The next criteria for classification is symmetry here. Now, one of the most important basis of classification is symmetry. Let's see how to study this symmetry first. So for studying symmetry, you should know about the axis. Some terms are there, you should know about it. So what is an axis? If you cut me from this sideways, will you find equal right and left halves? No. So if you want a particular left and right half of an individual, you have to cut it exactly from the middle portion, that is passing to the sagittal plane. Is it okay? So how you came to know it has to be passed through the particular plane? So that is because of this term, that is axis. This has been taken as an imaginary line. An imaginary line passing through the center of the body. Any organism's center of body, this line is passing, and that is what? The axis or the central axis. Now comes one more term which we used just now, that is plane. That also you should know. What is plane? Plane is any flat surface through which body is cut. Like how you will cut it. See, let's see. Suppose I cut this sponge. Now I'm cutting this sponge like this. Do you find equal left or right? No way. Okay, let's cut it from this plane, transverse plane. Do we find equal left or right? No. Even if you cut it like this also, you will not find any equal right or left halves. So that means if you pass any plane from this body, if you are cutting this animal from any plane, you are not getting equal halves. That is a perfect example of asymmetry found in sponges. But suppose if I cut this organism from one plane, that is a particular plane called as sagittal plane, which is passing through the center of the body. It is a sagittal plane which is just passing through the center like this. So definitely you will find what? Left and right equal halves. So that is an example of bilateral symmetry. But if you pass it from this end, if you cut it from this end like transverse plane, will you get half equal halves? If you cut it from transverse plane, will you get two equal halves? No. So that means one plane was only possible to cut this organism into two equal halves. That is the sagittal plane. So that one plane, when only one plane is possible to cut an organism into two equal halves, that is left and right, it is called as bilateral symmetry. But if you come to this case now, that is radial symmetry. In this case, if you take the plane, if you cut the organism from any plane, if you cut it from this plane, if you cut it from this plane, if you cut it from middle also, you will always get two equal halves. So now the radial symmetry you should understand, when an organism is cut from any plane and two equal halves are obtained. So it's called what? Radial symmetry. Is it clear? But when only one plane is possible to cut the organism into equal halves, it's bilateral symmetry. When no plane can cut it from into two equal halves, it is asymmetry. Is it okay? Now pentaradial symmetry is found in starfish, where you have only 5 angles, only 5 body parts can be cut. So you have only 5 planes possible to cut it. You don't have indefinite number of planes. You have only 5 planes to cut the organisms. That can be cut by only 5 planes. So let's talk about the next criteria for classification, that is segmentation. Now, do you have segmentation? Yes. You can see here, this is an individual who has 6 packs. So 6 packs which you develop, that is an perfect example of the symmetry, that is segmentation. Why? Because you also have some origin from fish-like organisms we have taken origin from. So that proves that segmentation is present in fishes, in the mussels, and you also have the segmentation. Okay. So this is something very important to understand first. So what is segmentation? Segmentation means when you have external and internal body parts correspondingly, making segments, then only it is called as true segmentation. That is called metamerism. So remember here, segmentation means the metamerism we are talking about, which is the true segmentation. Please understand very clearly. This is a true segmentation. Okay, what's that true segmentation? Again, I am telling you, when external and internal segments correspond to each other, that is called true segmentation. That is found in metamerism, is found in very important to understand. It is found in only 3 phylum. Can you see here? It is found in Annelida, Arthropoda and Chordata. Only 3 phylum. Please remember very clearly. See, it's very easy. This chapter is very easy if you just put little bit efforts. Here you see, this is an example of Nereis. So Nereis is what? Annelid. And this also is showing segmentation. Can you see? External and internal body parts, they are showing you the segmentation. That is an Annelid. So here it is Nereis. That is a perfect example of segmentation. Next comes your Arthropoda. This is a scorpion. So Arthropod also has what? Segmentation. External and internal segments correspond with each other. And next comes your human, that is chordates, or the fishes also. You can see they have all got segmentation. In segmentation, there is serial repetition of at least some organs in them. For example, if you talk about humans, don't they have segmentation in the vertebral column? Even they have segmentation in the muscles also and the nerves also. So serial repetition of at least some body parts should show in that segmentation. That will be always shown. In those segments, there will be serial repetition of at least some organs. That is written in NCERT also. Please highlight that point. Okay. So this is a perfect example of segmentation. But now here you can see one more, diagram here. That is pseudo segmentation. That means false segmentation. Why it is called false segmentation? Even you can see segmentation here also. No? This is the part called scolex. This is the neck, and the rest part is called as strobila. In the strobila, you can see some segmentations like immature proglottids here, mature proglottids, and the gravid proglottids. So still you can see some kind of segmentation externally. But here it is false segmentation. Externally you can see. Externally you're looking at someone, they're looking very beautiful, but may not be that much good. So in this case also, it is pseudo segmentation. Outside you can see, but they are not of true embryonic origin. So these segments are not of true embryonic origin. So they are not going to be considered as true segments. Is it okay with you? So example is Taenia solium or the tapeworm, in which you will see the pseudo segmentation. Clear, beta? Let's move further now. Next very important classification is the germ layers. Now these germ layers, from where you find these germ layers, where are you going to find these germ layers? These germ layers will be present in the embryonic stages. Like here you see zygote is undergoing cleavage, it is forming 8-cell stage, and then comes the blastula stage, and ultimately comes the gastrula. And just now I told you the gastrula has some opening here. This is called as blastopore here. This structure is called blastopore. This is called blastopore here, and this structure is called as archenteron, that is primitive gut. Archenteron is primitive gut. So now you see here, when this gastrula has formed, now it is going to differentiate further. So those differentiation will form 3 germ layers, that is ectoderm, endoderm, and mesoderm. Let's be very specific. So the 3 germ layers are the ectoderm, endoderm, and mesoderm. But here you see in this case diploblastic. So according to germ layers, there are 2 types of organisms. One is diploblastic, other is triploblastic. Let's see how. Very, very important here. In triploblastic, you have all 3 germ layers present: ectoderm, endoderm, and mesoderm. So you have all 3 germ layers present. All 3 germ layers present. What are those germ layers? Ectoderm, endoderm, and mesoderm. Please be very specific. All 3 germ layers are present. And what is the future of these germ layers? These germ layers are going to form your organs. For example, your heart is made from mesoderm. Your liver comes from endoderm. Your brain comes from ectoderm. So in that way, they have some fate maps by which they have studied what structure is going to form from a particular germ layer. Okay. So let's see now. In diploblastic, you have only 2 germ layers, not only 2 germ layers. Now, which germ layers are present in this? The only germ layer which is not present is mesoderm. So mesoderm is absent. Mesoderm is absent. So rest 2 germ layers you have present, that is ectoderm and endoderm are present. Here you see example. So example only 2, Cnidaria and Ctenophora. We don't consider the Porifera as diploblastic or triploblastic because they don't have true layers coming out. True germ layers are not present in them. Here you see next is your triploblastic. In triploblastic case, examples Platyhelminthes phylum 2, Platyhelminthes 2 chordates, all are what? Platyhelminthes till chordates. all are triploblastic. Is it clear to you now? Let's move further. And here one very important point. In diploblastic, you don't have mesoderm. Mesoderm absent, but one structure is always present, that is called mesoglia. So mesoglia present, you can write here. Mesoglia present, but mesoderm is absent. Mesoglia is present. What is mesoglia? Mesoglia is non-cellular layer. Mesoglia is non-cellular. This is non-cellular. So you can see here also, this doesn't have cells here. It is just having some fluid here, that is mesoglia. But in this case, the mesoderm, you can see particular cells also. But in this case, no cells. Is it clear, beta? So let's move further now. The sixth classification, the basis of classification is the coelom. Very, very important criteria for classifying organisms, because in that coelom only, that is body cavity, you will have the organs placed. So let's see what is coelom. Coelom is a body cavity. But one very, very important point, you should know what is true coelom. What is true coelom first? True coelom is always and always lined by mesoderm on either sides. Mesoderm on either sides. Okay, so it is lined by what? It is lined by mesoderm on either sides. Let's see how. Let's see once how. So here you see, this is a particular layer called mesoderm here. So this is a mesoderm. But still you can see the organism is acoelomate. That means it doesn't have coelom still, even in spite of having mesoderm. So if you talk about the Platyhelminthes, they have mesoderm, but still they are acoelomate. Anyway, the Cnidarians and Ctenophores, they have mesoderm. No, so they are not going to have what? Coelom anyway, because they don't have mesoderm. But in Platyhelminthes case, they have the mesoderm, still they don't have the coelom. Why? Because this mesoderm is solid. Mesoderm is solid and is filled inside the parenchyma tissue. So when it is filled inside, it is not forming a cavity. That is why it is acoelomate. In pseudocoelom, you see, pseudocoelom, there are three types of organism coming from the, because of the criteria that we have taken is coelom. That is pseudocoelom, means false cavity. The cavity has definitely developed, but that cavity is still what? Not lined by mesoderm. So it is false cavity. Third case, the coelom, that is a true coelom, is coming from the mesoderm. It is lined also by mesoderm. So this is a perfect example of classifying the organisms according to coelom. Even in pseudocoelom, the origin also of the coelom doesn't take place from the mesoderm. So let's see one point. True coelom arises from mesoderm layer. So true coelom comes from what? Mesoderm. But here you see, it is not coming from mesoderm. In pseudocoelom, it is coming from the blastocele that is present in the blastula stage, the cavity called blastocele. From that the coelom is coming. So it is what? Pseudocoelom. So let's see. So as per coelom, the organisms can be acoelomate, next is your pseudocoelomate, and the third organisms are your true coelomate, that is eucoelomate. Now these eucoelomates that is having true coelom can be further two types. One is schizocoelomate, which is not given in NCERT, but is still one point you should know. Okay, this is enterocoelomate. 1 minute, thus you see, it will take hardly a minute to understand this. The true coelom, that is eucoelom, is further classified into schizocoelom and enterocoelom. So what is schizocoelom and what is enterocoelom? during embryonic development, when the primitive gut was developing, that I told you, that is the archenteron. You can see here it's written also, archenteron. So this archenteron is the primitive gut. From that primitive gut, you can see there's a out pouch coming out. There's a pouch coming out, small pouch coming out, and this is a part of mesoderm which has come out. This is going to form a cavity. So this cavity which is coming from the enteron, that is primitive gut, will be called as enterocoelom. Enteron means gut. Coelom, which is coming from the primitive gut, that would be called what? Enterocoelom. Next, you see there is a cavity coming from what? The splitting of the mesoderm. There were some mesontoblast cells which gave rise to mesoderm, and this mesoderm has got a split now. So the splitting of that mesoderm gives a cavity, produces a cavity, and that is called as schizocoelom. Is it okay with you now? So let's see the different phylum in which what type of coelom is present. So let's see now. A coelomate, example only one, that is your Platyhelminthes. Platyhelminthes, only one example here. Pseudocoelomate, example only one again, that is Aschelminthes. Aschelminthes are the only ones which have the pseudocoelom. False coelom. Schizocoelom is present in, example is annelids, arthropods, and even you have the, just a minute, I'll remove this, annelids, arthropods, and you have the molluscans, and even the Aschelminthes also. Aschelminthes also have the schizocoelom. Next comes your enterocoelom. Enterocoelom is present in echinoderms, hemichordates, and chordates. Please write down this important point: hemichordata and chordata. Please write these important points. Okay. So let's move further and understand this last criteria for classification, that is basis of classification, that is your notochord. By notochord, you can easily classify the organism into two divisions. One is non-chordates, others are chordates. Let's see how. So this notochord is a solid structure. Can you see? This is a solid structure. This is mesodermal. First is mesodermal. That means it takes origin from mesoderm. Next, you see it is solid rod-like structure. Why you need a solid rod-like structure in a body? Because if you want to stand up, if you had only muscles, were you able to stand? You would have been just a sheet of somewhere, the sheet lying somewhere. So that means you should have some structure which makes you to stand erect, have some structure, that is your what? Rod-like structure called as vertebral column. So this vertebral column is developing from what? Notochord. Okay. So let's see. So this is mesodermal and taking origin from mesoderm, and there is rod-like structure which develops further into what? That is your vertebral column. Due to this notochord, the organisms can be of two types. One is the non-chordates which don't have the notochord, and the chordates which have notochord present. So notochord present here and notochord absent here. Now one question you tell me, one question I'll ask you. Notochord is absent. Now can you just recall the difference between the non-chordates and the invertebrates? Are all non-chordate invertebrates or all invertebrates are non-chordates? That one question comes to your mind, I think. The non-chordata. So are all invertebrates non-chordates or all non-chordates invertebrates? So see, non-chordates, if I talk about, these are what? These include phylum, these include phylum Porifera, phylum, members of phylum Porifera till Hemichordata. Till Hemichordata you have what? All of them are non-chordates. If you talk about chordates, now chordates are three types. There are three subphylum here. One is a Urochordata. Next one is your Protochordata. Sorry, it's Cephalochordata. Okay, the cephalochordates. Now you see, this is a Cephalochordata. So Cephalochordata and Urochordata together they are called as protochordates. The Urochordates and Cephalochordates together you call them as protochordates. And next is your vertebrates. Let's see now. If you see, the chordates include Urochordates and Cephalochordates, but they are not vertebrates, they are invertebrates. So these two are what? These two are protochordates and they are also invertebrates. So in this way we can't say that all invertebrates are non-chordates. We can only say all non-chordates are invertebrates. So please write down this point. All non-chordates are invertebrates. Please write down this point. Okay. Next question comes, are all chordates vertebrates or all vertebrates are chordates? So you can see all chordates can't be vertebrates. There are some are invertebrates also. So all vertebrates can be chordates. So this is the next point you should remember. That is your all vertebrates are chordates. So let me remove this first. All vertebrates are chordates. Hope you understood very well this classification now on the basis of notochord. So see, on some basis, some few criteria, you can classify the organisms very easily. This is a major classification which is required. Even minute classification also is done, but that is not required now at this stage. First, you should know about the basis of classification, how you can classify those majority of the animals. Is it okay? So let's continue further with the phylum Porifera, which is the first phylum in non-chordata. So why we call it as phylum Porifera? Because they have pores in their body. That is why it is called phylum Porifera. Phylum Porifera means they are pore bearers. Pore bearers. They have pores in their body. You can see here, these are small pores here. You can see these are small pores. So that is because of this they are called as phylum Porifera. Interestingly, they were earlier considered as plants. So scientist John Ellis gave them animal nature. Scientist John Ellis proved their animal nature. Very important. Proved their animal nature. This is not required more in your course, but little bit you should know about. Who gave the term Porifera? If I talk about, so term Porifera was given by Robert Grant. Few points you should know, not more. For example, common names. So this is a Euplectella. Euplectella common name you should know. It is Venus flower basket, which is given in Japan as marriage gift. Euplectella, there is a Venus flower basket. It is given in Japan as a marriage gift. Chalina is a dead man's finger. You can see a dead man's finger. It's appearing like that. So Chalina commonly called as dead man's finger. So Chalina and Clyona are two different terms. Please don't get confused. Most of the students get confused in this. Chalina is dead man's finger. Clyona is boring sponge. This is called boring sponge. So it is not boring that it is not interesting. In that way, it is not boring. It is boring sponge because it secretes some chemical by which they can, make those rocks also sometimes to break, because those chemicals, they can dissolve the part in that crevices they can produce in rocks, and the rocks can be cut in pieces. Even one very important question comes. This is the only sponge which is harmful to pearl industry, because this causes the oyster shell is there. It makes, the chemical makes some, it pores, it gets inside the oyster shell. It wants to feed on that, and it dissolves the shell part somewhere because of those chemicals. So it's a very dangerous, it is threat to pearl industry. So you can say Cliona is threat to pearl industry because it bores inside what? Bores inside the pearl oyster. As it bores through shell of oyster. Is it okay with you? Yeah, let's continue now. So next one, you see, this is a spongilla. This is given in NCERT also. Please remember this. This is a spongilla. Spongilla is a fresh water sponge. Fresh water sponge is spongilla. And the next one is your EU spongiae. So EU spongiae is called as bath sponge. EU spongiae, common name is bath sponge. So this is not too much required, but at least once you should go through it. Okay, so let's move to the general characters of Porifera. And you see habitat. They are mostly marine, but they are all aquatic because all live in water. So those all sponges you will find in what? Aquatic medium. Mostly they are marine. Mostly marine, but some are freshwater. Few are freshwater. Can you name one freshwater species? Just now we talked about, that is spongilla. Spongilla is freshwater species. Is it okay with you? Now, very important question comes. What is the level of organization? What type of level of organization is present in sponges? So it is just now studied cellular level only, because the body is made up of only cells. Next comes the symmetry. What type of symmetry they have? Mostly they are asymmetric, not all. Mostly asymmetrical, because some may have radial also. So mostly they are asymmetrical, you please remember. Next is digestion. Digestion is always and always intracellular. Please make it an important point here. This is an important point because this question is usually asked. So digestion in sponges is intracellular. It takes place inside the amoebocytes and those collar cells. Partial digestion takes place in collar cells and partial digestion takes place in amoebocytes. Please be very careful for this question. This question usually comes. Respiration is through body surface. This is through body surface. Okay. Next you see excretion is also through body surface. And product is ammonia. Ammonia excreted through body surface. Locomotion. No locomotion. This is absent here. Locomotion is absent in porifers. Skeletal system. Yes, they have some skeletal system. They have spicules. Spicules present, which help them in, forming some structure, because they are only cells. If they were only cells, how they could form a body? Because of those spicules, they form a particular shape. That is a type of skeletal system in them. Nervous system, there is no nervous system in them. It's absent completely. Now reproduction, usually it takes place by both asexual also and sexual also. Sexual by gametes and asexual by fragmentation or gemule formation. So reproduction is what? Both asexual or sexual. So it is asexual by fragmentation, and it is also by budding, that is gemule formation, that is internal budding. Gemule is internal budding. And the sexual process by gametes. Sexual by gametes. One very important question. Do they go for self reproduction? Because they are usually hermaphrodite. So sponges are hermaphrodite. Sponges are hermaphrodite, means bisexual. Hermaphrodite means bisexual or monoecious. Okay, so sponges are hermaphrodite. So question comes to you, this question will be asked, do they go for self fertilization or cross fertilization? So you have to answer they go for cross fertilization, because they are protogynous. That means female reproductive organs in them will develop earlier than male. So that is why they are called what? Protogynous, and they go for cross fertilization. So remember, even being hermaphrodite or bisexual, they have cross fertilization occurs. Cross fertilization occurs. Please remember this point. Okay. Now, last question, that is development is direct or indirect. So you remember development is indirect. Development is indirect. What that means? That means they have larval stage, larvae present. Okay, larvae is present. For example, the larvae of Leucosolenia is parenchyma, and the larvae of Sycon is amphiblastula. Please write down, larvae of Sycon is amphiblastula and larvae of the Leucosolenia is parenchyma. Please write it down. So let's continue further with the very important structure of that. Now, to understand phylum Porifera, to understand the sponge, you need to understand what is present in their body wall. How is the body wall existing in them? What kind of structures are present in them? Let us see. So in the body wall, you can see three layers first. Can you see these three layers? One is the choanoderm, that is innermost. In between you have the mesohyal, and the outermost is pinacoderm. So remember, pinacoderm is outermost. This is outermost. Choanoderm is innermost. Please remember this. This is innermost. This is inner layer. This is the outer layer. So pinacoderm is outer layer. Please remember, pinacoderm outermost. So what it includes, you can see there are some cells like pinacocytes. Can you see? This is a cell called pinacocyte. Flat cells called pinacocytes. Includes pinacocytes. Pinacocyte. These are flat cells. Flat cells. Even you can see some porocyte also. Porocyte is coming. This is a pore-like structure. This is a porocyte. So includes pinacocyte and porocyte also. So through the pores only the water will come. This is a channel protein-like structure through which the water has to come. The water is flowing through this porocyte. Can you see that? Let's continue further. So this is a pinacoderm. Next, middle you have the mesohyal. Please don't write the mesoglia. Don't write mesoglia. It is mesohyal. If you write mesoglia, it's wrong. They have mesohyal. And the mesohyal, it includes all the cells like amoebocytes, archaeocytes, okay, amoebocytes, archaeocytes, all these cells will be present, etc., etc. You can write the trophocytes, thesocytes, lophocyte, okay, archaeocyte, sclerocyte, oocyte, all these cells you will see in the mesohyal. And one by one, if you want to understand the function of them, you can see lophocyte. Lophocyte is involved in formation of collagen. So lophocyte forms collagen. Okay, oocyte, that is a female reproductive cell which will take part in the reproduction. Here you see the archaeocyte. Archaeocyte is totipotent cell. Please remember, this is a very important cell here. Archaeocyte. Archaeocyte is a totipotent cell. So archaeocyte is totipotent. What do you mean by totipotent? Totipotent means it can give rise to complete entire sponge. One cell can give rise to entire sponge. That is like a stem cell. So this stem cell can give rise to entire body, entire animal. That is archaeocyte is a totipotent. Sclerocyte. Can you see sclerocytes? Now sclerocytes you can see also. This sclerocyte is forming some structure here called spicule. So the sclerocytes are forming the spicules in the sponge. Clear beta? And you see now inside layer has choanoderm. Innermost layer is choanoderm, and the choanoderm is a characteristic cells are present in them, that is called collar cells, which are only found in what? The sponges. Collar cells are present in sponges and in proterospongea also are present, but mainly in animal kingdom. Now if I talk about, that is sponges, they are found in the sponges only. You see the collar cells which are called choanocytes, which help, they have flagella by which they can move the food also. You can see the food is trapped here and got inside the cell. Okay, so they can take the food from the water with the help of this flagella. Is it clear? So here inside you'll find what? Inside you'll find the choanocytes. On the inside layer you'll find the choanocytes. Is it clear? Let us continue further and understand more. Now you see water canal system is a very important feature of the sponges. Water canal system, which is a very important this characteristic feature of sponges. It is also called as aquiferous system. Now water canal system is of three types. Can you see? Ascon type, Sycon type and Leucon type. Ascon type, Sycon type and Leucon type. So water canal system helps in food gathering. It also helps in respiration and exchange of waste. Very important for your exams. Okay, the important functions, they will ask you what are the functions. So functions are food gathering, respiration and exchange of waste, or the removal of waste also you can say better. Okay, exchange of gases you can write, but removal of waste you can write here. So removal of waste you can say. Here you see beta, removal of waste. If you see three types of canal system are present: Ascon, Sycon and Leucon. There's particular difference in that. In Ascon, water moves through the dermal ostia inside the spongocoel and gets out of the osculum. So in Ascon type canal system, water moves into the ostia, that is the opening through which the water enters here, goes into the spongocoel, this is a spongocoel, and gets out of the osculum. This is the simplest type of canal system. Simplest. So this Ascon type is simplest type. Okay. Now we come to Sycon and Leucon. Much more complex than Ascon is Sycon, and the most complex will be the Leucon type. It's not there in a course, so don't worry. Nothing to worry. Just once you just listen, that's all. If the question comes, you'll be able to do it. Otherwise, leave this without any stress. So Sycon type, it has got some structure through which the water will move inside. That is prosopile. Can you see prosopile? Once the water goes to the dermal ostia, it goes into a structure called as incurrent canals, and then it goes to prosopile. Okay, afterwards it goes out into the radial canal, and through the radial canal they are going out through in the spongocele through a structure called the apopyle, and ultimately it will lead to the spongocele, that it will be thrown to the spongocele, and through the spongocele they are going to come out to the osculum. So it is dermal ostia, then incurrent canal, then it is prosopile, then radial canal, and then your apopyle, spongocele, and ultimately outside osculum. Don't worry, this is easy if you just have one mantra with you, that is dipraso. Let's see how. Dipraso. Dipraso is for Sycon type. Sycon type you can remember easily with this mantra, dipraso. D for dermal ostia, it is for dermal ostia. I for incurrent canal, P for prosopile, I will explain you, don't worry. R is for radial canal, A is for apopyle, S is for the spongocele, and O is for the osculum. So let's see how this mantra works here. It's very easy. You will never forget also. Let's see how. This is a structure called what? This is a structure here called as dermal ostia. So we can write here as dermal ostia because it is present in the outer part, and that's called dermis. So dermal ostia, or you can say ostium also, through which water enters now. So water has entered into what? It has entered into incurrent canal. It has entered into a canal called incurrent canal. So Sycon type, you see dipraso, D for dermal ostia, I for incurrent canal, it has gone into incurrent canal. From the incurrent canal it is coming into what? Radial canal. It is coming into radial canal. Through what structure? Through prosopile. So prosopile is leading into what? Prosopile is leading into radial canal. Prosopile is leading into radial canal. Is it clear now? After radial canal, this water is moving out through the apopyle into spongocele. It is moving into what? Spongocele through the apopyle, and from the spongocele it is going out to the osculum. Okay, is it clear to you? Don't worry, only once you have to listen. Don't worry about more complexity, because even further we have one more complexity called leucon type. That is not in your course. I am not going to explain you because it becomes tough for you. Only one thing you remember, that has one extra thing called excurrent canal. One extra structure called excurrent canal has developed in that. That's it. Okay. Rest all remains same. Dipraso is there, but if one extra thing came out, that is excurrent canal through which it is going into the spongocele. Okay. Don't get confused. Don't take stress. If you can do this, it's okay. Otherwise, leave it. It's not going to come in your exam. Just for your, maybe a question comes, one question also, if it comes outside syllabus, you should be able to do it. That's it. Next criteria we'll talk about, next, a special character we'll talk about is spicules, which are very helpful to they are a part of a skeletal system in sponges. They make the sponge to stand erect. Means if they are standing at some place, they have to stand, or they have to, structure has to be maintained. So that structure has to be maintained because of what? Because of spicules, correct? Now different types of classes are given here. You can see Calcarea is one class. There is one class called Hexatenida, then Demospongia. There are three classes. So nothing to worry about further classification of sponges. Don't worry, but you should just remember there are three classes. One is Calcarea, Hexatenida, and Demospongia. Suppose you want to classify the mammals, you have some orders like Perissodactyla. Okay, you have Edentata, you have the Cetacea. These are orders. Okay, so we are not going to worry about more about them. Here you see the Calcarea class will have the spicules which are calcareous spicules, means they are made up of calcium carbonate. Spicules made up of calcium carbonate are the calcareous spicules. The spicules made up of silica, that is SiO3, I think, that is silica. So the spicules which are made up of silica are siliceous spicules. Here also in Demospongia also there are siliceous spicules, but in Demospongia they may have some protein fibers also. Spongian is a protein fiber, so they may have protein fiber also. Is it okay? Now you see, or even the Demospongia may have both also, because sometimes if it is only made up of a spongian, it is very soft like bath sponge. So they may have spicules, little bit spicules, siliceous spicules in that to make them little bit strong. So let's see, Hexatenanida. In this case, you see there are 6 rayed spicules. Only you have to remember hexa, 6, tena means rays. So 6 rayed spicules you can see, 6 rayed spicules. Okay, so they have 6 rayed spicules. So Hexatenanida class has 6 rayed spicules. Just you remember this. 6 rayed spicules like this. 1 ray, 2 ray coming like this. 3, 4, 5, 6. Like this you have 6 rayed spicules. Is it okay with you? Is it okay? Yeah. It's very easy. Don't worry. And don't think it is tough for you. It will become definitely tough if you think that it is tough or it's boring. Actually, it's a scoring. It's scoring. You have to score. In NEET exam, you have some questions like last year, 5 questions were asked from this. So 5 questions, or 4 questions make a big difference. Okay. So let's continue with further one special feature of Porifera, that is gemule formation. Now, what is gemule formation? Gemules are, these are internal buds. These are internal buds. What do you mean by internal buds? Why they require internal budding? In sometimes during unfavorable conditions, they form gemules. So gemules are formed during unfavorable conditions. Formed during unfavorable conditions. Very, very important point. Usually the freshwater sponges, they go for gemule formation, but even marine water sponges also go for gemule formation. That is not only freshwater, but mainly freshwater will definitely go, because there are more unfavorable conditions in freshwater as compared to the warmer regions. Yes, see. So gemule formation, what happens? When the unfavorable condition will arise, they will collect all those archaeocytes. Remember, archaeocytes were totipotent. Now, for example, any threat comes to you also, what you do is you will collect all the valuable things and then you will run with those valuable things, correct? Same thing happens here also. The most valuable thing is what? Archaeocyte. It is a totipotent cell. So that totipotent cells all will be taken together, covered by a membranous structure. This membranous structure will have some monoaxon spicules, to make them stronger outside. So that double layered structure is there now, and it has monoaxon spicules inside, which give a protection. When the favorable conditions arise, so these archaeocytes, they are going to come out through a structure called micropyle. Through micropyle, this will open. This is a operculum over this. So this operculum or the lid will open, and the archaeocytes are going to come out of the micropyle and give rise to each and every one archaeocyte is going to give rise to new sponge. Is it okay? So it is a type of internal budding, please remember. But one very important question comes. How is this gemule formation different from formation of reduction bodies? So what are reduction bodies? These are reduction bodies here. So reduction bodies What are these reduction bodies? Difference you can see. Difference you can easily see here. Here you see it is a amoebocyte. It is a amoebocyte instead of archaeocyte. Instead of archaeocyte, here you find amoebocyte. And it is not a internal budding. It is not a type of internal budding. Not a type of internal budding. Then what it is? Sir, then what it is? Okay, let's see. Now suppose this is also formed during unfavorable conditions. First you remember this also. Okay, let's talk about this. Formed during unfavorable conditions. What are these then? What are these going to do now? So in the unfavorable conditions, what they are going to do, they are reducing in size. That is why reduction bodies. Please remember, reduction word is there in that. So if someone asks you a difference between gemule formation and reduction bodies, it's very easy. In reduction bodies, the size is going to reduce. Okay, during unfavorable condition, the sponges reduce in size. Reduce in size and shrink. Okay, and what they do, they cover those amoebocytes now. The amoebocytes are covered with pinacoderm layer. So amoebocytes are covered with pinacoderm. So when the conditions when the conditions will become good, these amoebocytes will develop further into other type of cells. Even these amoebocytes can develop into archaeocytes also. Okay, so let's continue further now. I hope you understood this difference between the gemule formation and reduction bodies. Okay, so let's further move with the next phylum, that is Cnidaria. Cnidaria, old term was Coelenterata. Why this name was taken out? Because coelenteron, that the word came because of coelenteron, that is gastrovascular cavity. So coelenterata word or the term came because of coelenteron. What is coelenteron? That is called gastrovascular cavity. They thought that all have gastrovascular cavity. First it was thought that all have gastrovascular cavity, but it's not correct. Let's see how. So the gastrovascular cavity, if you see, this is the hydra here, and this hydra will have these structures. This is the hydra inside a structure. Okay, yeah. So this is the gastrovascular cavity. Can you see? This is the cavity inside. So this cavity is called as gastrovascular cavity. As the word says, gastro involved in digestion also. Vascular, that means circulation. That means whatever the content is digested, it has to be circulated to the cells. So that is why it is called as gastrovascular cavity. Earlier it was thought it is present in all types of coelentrates, but it is not found in all types of coelentrates. That's why the new term came called Cnidaria. So this term was given by Hatschek. The term was given by Hatschek, and that means presence of Cnidaria word came due to the presence of cnidoblasts. Presence of cnidoblast cells. What are cnidoblast cells? Cnidoblast cells means stinging cells. So those who have those stinging cells, they will be called as cnidarians. Is it okay with you? Now let's see. This is structure. Some students, they call this structure, you can say, as mouth oblique anus. Correct? This is the structure which is called mouth oblique anus. But here you see one more structure is there nearby. This one. This is not mouth. This is called as hypostome. Please be very specific. Hypostome is not mouth. It is a structure surrounding the mouth. So hypostome is the structure surrounding the mouth. Okay, don't confuse with that. Now let's talk about the common names of these animals, that is Physalia. Physalia is called as Portuguese man o' war. Okay, Portuguese man o' war. Gorgonia is called as sea fan. Can you see? It is called sea fan. You have to remember these names. Very, very important. You have to remember. You just don't have to listen to only lectures. You have to remember them. Sea fan. So you have to just representation. If I talk about it is like this. Sea pen. You see, pen is given here only. Pen. So if they ask you the scientific name of sea pen, it is clearly given in that only. It's very easy. Sea pen is Penatula. Sea fan is Gorgonia. Alcyonium is the dead man's finger. Here also it's called as dead man's finger. So dead man's finger of the Cnidarians is Alcyonium, and dead man's finger of Porifera was Chalina. Corallium. Corallium is the red coral. Please write down red coral. Now in the chat box, can you write which is blue coral? Yes, many types of corals. Blue coral, okay, staghorn coral. There are many types of corals, but you don't have the all these things in your syllabus. That's why I'm not going ahead. Okay, but anyway, Heliopora is called as blue coral. I think just check it. Cnidaria. So other important, common names we'll see. Meandrina is called as brain coral. Adamsia is sea anemone. Sea anemone is Adamsia. Aurelia is jellyfish. Please write down all these points. Jellyfish. Hydra. Hydra is the freshwater inhabitant. It is found in fresh water. Mostly the sponges, the others, Cnidarians are marine, but Hydra is freshwater. Is it okay? So Cnidarians are characterized by two structures, that is polyp and medusa. So what are polyp and medusa? Medusa is mouth down, umbrella like structure you'll see. This is the medusa. Okay, so this is the medusa here of the jellyfish. So medusa is this one. This is mouth down, and polyp, if I talk about, this is the polyp here. This is the Adamsia, and you can see the Adamsia. The mouth is up here. So mouth up is the polyp. Polyp is mouth up. Okay, polyp always goes for asexual reproduction. And if I talk about medusa, it is mouth down. And here you have sexual reproduction going. Is it clear with you? Done? Let's continue further now. Now some general characters we'll talk about. That is habitat. Habitat, if you see, they are mostly, all are aquatic again. All are aquatic. They are mainly marine. Mostly they are marine. Few are freshwater. Few are fresh water, like Hydra. Just now I told you. Level of organization is tissue level. Tissue level of organization. Symmetry is radial symmetry here. So you have radial symmetry. Germ layers, according to germ layers, it is diploblastic organism. Why? Because it has two germ layers. One is endoderm, other is ectoderm. They don't have mesoderm, they have mesoglia inside. Digestion, both extracellular and intracellular. So what do you mean by this? Now, first the food will come in the cavity, that is the cylindron, and then the further digestion will take place in the cells. So here you see, first extracellular, you have to write here, first extracellular digestion, and then intracellular digestion. Is it okay, beta? Is it okay? Clear? Excretion now. Excretion by body surface again. By body surface. Nervous system. Yes. The first time in evolution, nervous system appeared for the first time in Cnidarians. Nervous system appeared for the first time. Appeared for the first time. Is it okay? Nervous system appeared for the first time in Cnidarians. So that means nerves arose first time in which phylum? If the question comes nerves arose in which phylum, then it is not Cnidaria. Neurons developed in phylum, which phylum they developed first time? That is Cnidaria, correct. Nervous system appeared for the first time in Cnidarians. Neurons appeared first time in Cnidarians. But if the question comes nerves arose first in which phylum, then it is not Cnidarians. So please write in the chat box in which phylum did the nerves appeared for the first time. Nerves are actually bundle of neurons packed by connective tissues. Okay, so write in the chat box. I'll check it. In evolution, nervous system appeared for the first time in, and here they have apolar neurons. Which type of neurons they have? Apolar neurons present. If the question comes, which type of nervous system is present in Cnidarians? It is diffuse type. Nervous system is diffuse type. That means they don't have proper orientation from where the signal is coming. So this type of nervous system is called what? Diffuse type. They don't know from where the signal is coming properly. So that is why it is called diffuse type. Clear? Reproduction now. So they reproduce by usually by sexual process. But in polyp it's asexual. So polyp has asexual reproduction. And medusa reproduces by sexual reproduction. Is it clear with you? Medusa reproduces by sexual means and the polyp reproduces by asexual means. Clear to you? Let's move. Reproduction and moreover development is indirect in them. They have larval stages also. Okay, let's move to the very important structure in Cnidarians, that is the presence of cnidoblast. So cnidoblast is also called as cnidocyte. Cnidoblast is also called cnidocyte. It includes nematocyst. Okay, it includes nematocyst. Nematocyst is present in them inside. Okay, nematocyst present inside the cnidocyte. Please write down nematocyst is stinging capsule. This is a stinging capsule present inside the cnidoblast. Hope it is okay with you. Clear? Let's see now how this is the undischarged one. This is undischarged cnidoblast and this is the discharged one. So what is happening here, you see, this is NCERT diagram that also is shown to you so that you can understand a little bit. Here you see this is undischarged state. This has a structure called cnidocyll. So cnidocyll is a trigger. So suppose in the near the body, if the prey is coming to near the body, so what will happen? This cnidocyll will be triggered and this will cause the passing of the signal inside, and this is structure which is there inside nematocyst will come out, ooze out, and this is coming up like this is bursting like this, and here you can see this is coming and it has some spines. It has very small spines here and there's a tube coming out, and this tube may have opening also through which hypnotoxin may be released. Is it okay? Or this tube can be used to encircle the prey. It can just hold the prey with that. So there are so cnidoblasts present on the surface of the tentacles. Tentacles have they are called battery of cnidoblast because they have so many of them on the tentacles. Okay, outer surface of the body also has, but maximum are found on the tentacles. So can you see this structure has come out and it is used for what? It is used for anchorage. What is the function also they may ask you. So let's talk about the function. Function of cnidoblast. Cnidoblast function. So it is anchorage. Anchorage, they can, get in some place, particular place. If they want to stand or something, they can anchor by that. So it is anchorage. Next is defense purpose also they can use, and capturing prey also they can use. For capturing the prey also they can use. So this is important question they may ask you sometime. Is it clear now? Easy for you now? Let's talk about one very important event going on in some cnidarians, that is called metagenesis, which is also called as alternation of generation. Metagenesis is also called alternation of generation. What do you mean by alternation of generation? How is it different from the one which is found in plants? This alternation of generation, okay, is different from the one present in plants. How you can say now? Let's see how. Very important to understand. In alternation of generation in plants, one phase was haploid, other was diploid. But here there are two phases, that is polyp and medusa. Both are diploid. You can see here. Medusa also diploid. Even the polyp. Okay, this polyp is also what? Diploid. So this polyp is also diploid. Even the medusa is also diploid. Only and only during the sexual phase they will produce some gametes. That's it. Otherwise, entire cycle you can see it's diploid. Red color you can see here. Red color is diploid. Can you see? A mitosis is going here. Even you can see here also mitosis is going. So polyp and medusa both are diploid. Both are diploid. Please write down this point. Both are diploid. Hope you understood this point very clearly. Now, what is happening in the life cycle of Obelia? Now, this is Obelia. Obelia is commonly called as sea fur. In the sea fur, you can see this is Obelia. So, what is happening? This is the colony of Obelia. Question also came. Colony of Obelia. Obelia is trimorphic colony. Obelia has trimorphic colony. What do you mean by this? It has trimorphic colony. What do you mean by this now? Trimorphic colony means it has three types of zooids. One is the blastostyle, other is the polyp, and one more is the medusa here. So they have three polyps. Trimorphic colony means three types of zooids. They have three types of zooids. What are those three types of zooids I told you? One is the polyp. Okay, one is polyp. Other is blastostyle. And the third one is your medusa. So three types of zooids you are finding. In this three types of zooids. So this blastostyle is a reproductive polyp. You can see this is producing medusa. So this is medusa. Now this medusa, the male and female will produce the gametes, that is egg and sperm, and they will form a zygote. So this zygote undergoes mitosis and forms a planula larvae. This is a larvae here, which is undergoing division and forms a polyp, and this polyp makes a branching colony here now. So it has three types of zooids. Is it clear to you now how this Obelia divides? Obelia has medusa and polyp. Medusa undergoes sexual reproduction. So here it is what? Sexual reproduction. Polyp undergoes what? Asexual reproduction. Polyp undergoes asexual reproduction. Hope this is very clear now. Okay, and both are diploid here. I told you both are diploid here. Is it clear? So let's see one more special character of Cnidarians, that is some Cnidarians they form corals also. So some Cnidarians are involved in coral formation. Some Cnidarians involved in coral formation, and very important, involved in coral formation. How these corals are formed? You see here. Can you see this volcanic island is developing here in the sea? Volcano has erupted, and this volcano is erupting still. So lava is coming here, and lava is solidifying. You can see here, lava is solidifying here. So when the lava solidifies, doesn't it form a structure outside that volcano also, outside the hill type of structure? Nearby the hill structure, you'll find some lava collecting in the bottom. So this lava is going to collect here. After that, you can see there's a cavity being formed, means there's a area which is forming. Here the fresh water by the rain can come, and there you can find some water is collected here, and this water is forming a body here, water body, and surrounding in the water body you can find some corals are developing here. A coral island develops here. So when it is very near to the this structure, the body, the mountain, so it is called as fringing reef. But when it is far from that area, it is very far from that shore region, so that is called as barrier reef. For example, Great Barrier Reef of Australia, that is an example of barrier reef. It is very far from the shore. Okay, and there you see there the water body also, which is bigger water body here, called as lagoon. But in some cases, when the volcano has resided, can you see the volcano island has completely resided, so still the coral island will be there, and that coral island is along almost a horseshoe shaped structure, and you can see water body is entirely present, completely present here. Yes, they definitely have some channels through which seawater can enter, that is there always, but this water body is called as lagoon. So this is an example of atoll which is there in the Andaman Nicobar Islands. Is it okay now? Is it clear with you? Let's continue. The next phylum, that is Ctenophora. Now Ctenophora, they are characterized by bioluminescence. They are commonly called as sea walnuts. Commonly called sea walnuts. Sea walnuts, sea gooseberries, and comb jellies, anything you can call. Okay, so they are commonly called as sea gooseberries, sea walnuts, comb jellies. Here you see some examples: Pleurobrachia, Ctenoplana, and Baroe. Can you see in the night they are glowing? So they are exhibiting bioluminescence. They are characterized by some comb plates. Can you see these comb plates here? These are comb plates. So if the question comes, how many rows of comb plates are present? It is 8 rows of comb plates. And they have tentacles also. Okay, if the tentacles are present, they belong to Tentaculata. If they don't have tentacles, they belong to a group called Nuda. So mostly they have tentacles in them. So this is Pleurobrachia, but you see Baroe doesn't have tentacles. Okay, so let's see the main characters of that, some general characters of phylum Ctenophora. They are very similar to the Cnidarians. Let's see how. Habitat, they are exclusively marine. So they are exclusively marine. They are not found in fresh water or somewhere. They are found in marine waters. Level of organization, again it is tissue level. Symmetry again is radial, because they are very similar to the Cnidarians. Germ layers, according to germ layers, they are diploblastic organism, means they have 2 germ layers. Digestion same as Cnidarians, extra and intracellular. Extra and intracellular here again. Is it okay? Excretion. So the excretion is, there's no specific excretory organ in them. By body surface, ammonia is released. By body surface. Nervous system, diffuse type, as in the Cnidarians, because they're very similar to Cnidarians. Now reproduction, only by sexual means. Only by sexual means. So they only reproduce by sexual means. They are hermaphrodite. They are hermaphrodite. That means they are bisexual. All are bisexual here. So sexes are not separate. Is it okay? Development is indirect. Development is indirect. They have a larvae called cydipid. Larvae is cydipid. Okay. And the development is indirect here. Fertilization is external. Fertilization is external. Please write down these points. Is it okay? Examples already we discussed. Some more important points. 8 rows of comb plates. Coloblast present. They don't have cnidoblast, they have coloblast present. Exhibit bioluminescence. Please write down. Exhibit bioluminescence. So they exhibit bioluminescence. Is it clear with you? Let's continue further. The one very important point, that they don't have any polyp phase, they have medusa phase only. Medusa only. Medusa present only. They don't have any polyp phase in this. Is it clear? You can see all these organisms, they don't have any polyp phase. They have medusa phase only. So is it clear till now? So let's continue with the phylum, that is Platyhelminthes. Now Platyhelminthes are commonly called as flatworms. Platyhelminthes are commonly called as flatworms. They are dorso-ventrally flattened. Please write down. Dorso-ventrally flattened. They are what? Dorso-ventrally flattened. Is it okay? Here you can see this is the fasciola, the common liver fluke. The liver fluke is fasciola. Schistosoma is called as blood fluke. Can you see here? Blood fluke. So blood fluke is schistosoma. And you can see in schistosoma, it is the one platyhelminth which is dioecious. Male and female you can see separate. Female is inside the gynecophoric canal of male. Here is Echinococcus, that is dog tapeworm. Echinococcus is called what? Dog tapeworm. Dog tapeworm. This is the smallest one. And Planaria is also called Dugacia. It shows excellent regeneration. This shows very good regeneration. So Planaria exhibits regeneration. Very high regeneration you can find in Planaria. Liver fluke causes a disease, okay, in sheep. Schistosoma is the human blood fluke. Schistosoma is the human blood fluke. Here you can see here. Schistosoma is human blood fluke. In human blood fluke, I told you it is the dioecious organism. This is only dioecious here. It is dioecious. Male and female you can find separate. Okay. And female resides inside the gut of male. Okay. Better you can write inside the gynecophoric canal of male. Inside the gynecophoric canal of male. Is it clear with you? Is it clear now? Gynecophoric canal of male will have the female inside. This is very clear. You can see here in the diagram. Is it okay? Let's move further. Even one very important structure is given as tinea solium. So tinea solium, I told you, it has 3 parts. One is the scolex, the neck, and the strobila. So strobila includes immature proglottids, mature proglottids, and gravid proglottids. In the gravid proglottids, you can see there is a complete uterine branching. Uterine branches, you can see uterus has got complete branching in that. So this is called as gravid proglottid. This is the Gravid proglottid. So gravid proglottid is characterized by branching, uterine branching. Okay, here it is uterine branches coming out. In mature proglottid, you can find both type of reproductive systems, male and female. But immature, you won't find the development of both type of reproductive systems. Is it okay? Let's continue further. What are the general characters we'll talk about? Habitat. So mostly endoparasites. Mostly endoparasites. And here you see level of organization. So they have now a better organization, that is organ system level. Correct? Symmetry. They have bilateral symmetry. Germ layers, they are the first triploblastic organisms. Please write down. Triploblastic. They are the first triploblastic organisms. Coelom is absent. They are acoelomate. They are acoelomate. Please write down this point. They are acoelomate. Segmentation absent. They don't have any segmentation in body. Digestion, they have particular blind sac body plan. So they have blind sac body plan. That means they have incomplete digestive system. Incomplete digestive system. That's called incomplete. If they don't have both opening, it's called incomplete. Excretion. Excretion is by special cells called flame cells. So flame cells or the solenocytes are involved in the excretion process. Nervous system is ladder-like. Nervous system is ladder-like, having some ganglia and double ventral nerve cord is there. So lateral nerves will join the ganglia. So they will have ladder-like nervous system. Reproduction. Reproduction is by sexual means and fertilization is internal. So here you see fertilization is internal. Fertilization is internal. Fertilization you can find is internal in that. Development is indirect. Development is indirect. That means larvae are present here. Is it okay with you? And reproduction by sexual means usually. In some cases they may have fragmentation also. Life cycle of fasciola. Now very important. It is not in your course, yes, but it's still 1 point, 1 minute it will take to just have a glimpse of that if the question comes. So let's see how to remember it easily. Remember MSR CM. Mr. Madhusudan Reddy is going to be a CM of Hyderabad or Andhra Pradesh. So let's see. M is for Miracidium. You have to remember like this. That's it. Miracidium. This is the sporocyst larvae. This is the rhedia larvae. This is the Cercaria. All these things you can see here. This is the Metacercaria. So these are the larval stages in the life cycle of Fasciola hepatica. You can see this is the Fasciola hepatica. Larvae are being produced. This is the Miracidium larvae. So Miracidium larvae, this infects snail. This infects snail. Metacercaria infects the sheep. Okay, or the goat. Clear? So let's see how. Let's see how it is going. So this sheep, through the feces of the sheep, the larvae has got into the pond. Miracidium larvae gets into the pond and it starts swimming now. After swimming, it bores inside the snail's body, enters into the snail's body, converts into a next larvae that is called a sporocyst larvae. So Miracidium converts into a sporocyst now. So this sporocyst inside the body of the snail converts into a next larvae by a process called as the pedogenesis, that is converting into rhedia larvae. Next, the rhedia larvae now converts into a other larvae called Cercaria, again by pedogenesis. So rhedia larvae converts into a Cercaria larvae, which starts now a little bit moving and comes out of the body of the snail, converts into the next type of larvae that is called Metacercaria, and this metacercaria is infective to sheep. So this metacercaria, which is there on the leaves in the pond, sort of in the shore of the pond, have some, the banks of the pond have some leaves, and they get encysted in that. This is an encysted form that can remain for many days. Afterwards, when these leaves are eaten by the sheep or goat, they get transferred in them, and they get into their bile duct. So they reside actually in the bile duct of the sheep. So understood very well. This is MSRCM. This is how you will remember this life cycle. Easy for you. Let's see some very important adaptations. These platyhelminthes, they are very smart. They have some parasitic adaptations by which they will not make the host to die also. No parasite wants that the host should die. And in that way, they have some very special mechanisms to protect themselves also, and not to have any effect from the immunity of the host. Immune system of the host also they can dodge. Let's see how. They have very thick body covering made up of cuticle. So this cuticle will prevent them from the digestive juices. Prevents from digestive juices. Are you getting point? Digestive juices or enzymes of the host. So very important. First point you saw, there was a covering over the body. Next is the presence of suckers and hooks. So why they need suckers and hook? Because now the food is flowing inside the intestine. So they should not get flowed down. They should not get carried away by that flow. So they should have some structure by which they can attach to the intestine wall. And for that they have very special structures called suckers and hooks, by which they attach to the intestine wall. Got it? Loss of locomotory organs. They don't have any locomotory organs. So that's a parasitic adaptation, because if they would have started locomoting, the immune system would have dodged. They would have tried to kill them. But they are just flowing with the stream, with the body fluids. So immune system also will not recognize them as a threat also. Absence of digestive organs. In some cases, like in tapeworm, they have complete absence of digestive organs. So they just have to suck the blood or the digestive food, and they are growing in size. So they don't have any development inside of organs. So that's very good for them. And they go for anaerobic respiration. Some have anaerobic respiration, so they don't have to come out of the body. So they are very good endoparasites. Well developed reproductive system. Now you see, in that case what happens? If they die, so their progeny should continue. So they have very well developed reproductive system. They produce eggs in lakhs and lakhs. So in the outside there's a threat. Outside definitely there will be threat. And how many will survive, we don't know. So that is a phenomena in them to continue their species on this earth. They keep on reproducing in large numbers. So when they reproduce in large numbers, the survival rate is more. Okay. So let's see now the roundworms. The next phylum is your roundworms here. Roundworms, that is Aschelminthes. So Aschelminthes are commonly called as roundworms. Ascaris is the common roundworm here. Common roundworm is your Ascaris. Wuchereria is your filarial worm. This is the filarial worm. Please remember, this is filarial worm, which causes filariasis or the elephantiasis. This is the Anthrobius. Anthrobius is the pinworm. Okay. This is the Rhabditis. This is semi-parasitic. It is not completely parasitic. This is semi-parasitic. Okay. And Xylostoma is a hookworm. This is called as hookworm. And Melodyne incognita. This is very important. You study in biotechnology chapter. So this Melodyne incognita causes the problem in the roots of the plants. So remember, Aschelminthes are parasitic not only on animals, they are parasitic on plants also. So this causes some gall, root disease in the plants. So Melodyne incognita can cause some, diseases in the roots of the plant. They usually reside there only. Okay, so let's talk about the further general characters of Aschelminthes. The habitat, again, they are mostly what? Endoparasites. So you can say parasites better, not endoparasites only. They are sometimes outside also. Parasites. Parasites not only on plants, they are also on animals. So parasites on plants and animals both. Level of organization I talked about was organ system level. If you remember, we just now talked about organ system level. Symmetry, again, it is bilateral here. Even they appear circular in structure, but still they have bilateral symmetry. Germ layers, they are triploblastic here. Triploblastic, they have all three germ layers. Coelom, they have pseudocoelom. They are characterized by false coelom developing from blastocele. So they are pseudocoelomate. They are pseudocoelomate. Segmentation, no segmentation. It is absent here. No segmentation. Please remember, digestion, they have very well developed muscular pharynx by which they can suck the blood or something. So they have well developed muscular pharynx, and digestive system is complete. Well developed muscular pharynx, and they have complete digestive system. They have complete digestive system. Hope you are getting my point. Let's see. Circulation. They don't have any special circulatory system, but yes, they have pseudocoelomic fluid in that, which helps in the passing of the content, which is digested content. Excretion. Excretion is by H-shaped rennet gland. H-shaped rennet glands. And the product comes out of the body by excretory pore. So there's an excretory pore by which the waste comes out of the body. Reproduction, if I talk about, only by sexual means. By sexual means. They are mostly dioecious. They are what? Dioecious. So nematodes are dioecious. Please write down. These are dioecious. In this case, very important, the females are longer as compared to males. Females longer as compared to males. Is it okay? Clear to you? So let's continue further with the next phylum now. With a very important life cycle of Ascaris. One very special character we'll study is life cycle of Ascaris. Again, you don't worry about the complexity of that. Just see little bit, what goes on in your body when the Ascaris infects. How the Ascariasis spreads. Just see once. That's it. Let's see now. The fertilized eggs are released into the soil. Now you see, this Ascaris, male and female, were living inside the intestine. Can you see this is the intestine of human being? The male and female are living here. Male and female Ascaris are residing inside the intestine. Male and female are residing, Ascaris residing in intestine of human. Okay. So let's see what happens. They copulate and they produce eggs. These eggs are released in the feces. Through the feces, they are released in the soil. So you can see the first stage larvae is still in the soil. The second stage larvae is also in the soil. So these are called as embryonated eggs. In the second stage larvae condition, they are called as embryonated egg, which are swallowed by sometimes by humans through the contaminated food and water. Please remember, they are swallowed by the humans through contaminated food and water. Now what happened? That second stage larva has entered into humans. Clear? Let's see how. So first stage juvenile is called as rhabdithiform larvae. So this first stage larvae is called rhabdithiform larvae, which undergoes molting. Second stage juvenile is produced, which is present here as embryonated egg. So this is the embryonated egg and this is the rhabdithiform larvae. Okay? Let's see now. These embryonated eggs are swallowed accidentally or by contaminated food and water and enter in the enter in the body of human beings. So they bore through the intestinal wall and go into the circulation. Now next you see it goes into the mouth, through the mouth it reaches the intestine, and this second stage larva hatches out here. It hatches out in intestine and goes into the hepatic circulation. Through the hepatic portal system it enters the liver. Okay, so through the hepatic portal system it enters the liver. In the liver it starts a little bit growing. After that, through the liver, through the inferior vena cava, the blood is flowing. From the liver it enters the heart, and heart is pumping the blood, and when the heart is pumping the blood to the inferior vena cava, it is entering the heart, and the blood has to go into the pulmonary circulation. So with the pulmonary circulation, this reaches the lungs portion now. So in the lungs portion, which stage larva has reached? That is the third stage larvae. So third stage larvae and the fourth stage larvae is found in the lung region. Clear? Now in the lung region they molt once and then they reside there in the alveolar region. Afterwards they sometimes accidentally, when you sneeze or when you are coughing, so what happens that through the cough very small larvae they enter again into your intestine. Okay, again through there's a common passage to the pharynx. So what they do is they again enter into the intestine. Afterwards in the intestine they grow in size. The fourth stage larvae, they grow in size and they become a adult stage. This is the adult stage, which is again here now. The male and female will again copulate in the intestine. So you saw that there was no other host for this organism. Only the humans are the ones which are being infected. Is it clear? So you understood very clearly how it happened. First stage larvae, this is called rhabditiform larvae. Second stage larvae, this is called embryonated egg, which are swallowed by them, and then they enter in the intestine. To the intestine they go into the hepatic portal system. To the hepatic portal system they go into liver, and ultimately they go into the inferior vena cava and reach the heart. Through the heart again the blood is pumped by the pulmonary circulation to lungs, from where they start molting. Third stage and fourth stage larvae is present there, and after molting they go into the intestine from the lungs, and there the adult will reside. So adult is residing inside the intestine now. So there will be many of them, and they will copulate inside and produce eggs again. Is it okay? Let's continue further now. So all these things I have explained you. Just you have to look into that, how the spread is happening. Okay. So in the lungs you can see the second and third juvenile stage is there, which is going for molting. So second molting and third molting will take place in the lung region only. After that the third stage juvenile enters the pharynx and it is entering the intestine. Okay. The fourth stage juvenile is entering the pharynx and ultimately coming to the intestine. Okay. So let's continue with the next phylum now. So next phylum is your Annelida. So let's continue with this Annelida now. So annelids are characterized by segmentation, and few common names of annelids are sea mouse. Sea mouse is the Aphrodite. Pheretima is the common earthworm. This is the earthworm. Lumbricus is a European earthworm. This is the European earthworm. This is European earthworm. Yes, this is Nereis. This is marine here. This is marine, and here it is very important to understand this is dioecious also. Okay. Hirudinaria is a common Indian cattle leech. Common Indian cattle leech. Please remember, very important, because some students they get confused. You can see that they are looking same here. They are looking almost same, but some regard both as dangerous leech, but both are not dangerous. This is very good for your health. This is used for taking out the impure blood from your body. There is a science called phlebotomy in which they take out the impure blood. So this is a medicinal leech. This is medicinal leech. It is not harmful for you. It is very beneficial for you. So see the structure diagram properly. It is hirudo, and this is Indian cattle leech, which is dangerous, which sucks the blood because it produces an anticoagulant called hirudin. So hirudin is an anticoagulant that will not make you realize also your blood is being taken somewhere. Okay, when it sucks the blood from that region. So let's see now some special general characters of annelids. So some general characters, if I talk about their habitat, so they can be terrestrial also and aquatic also. Habitat, if you talk about terrestrial and aquatic. Nereis is aquatic, Nereis is aquatic. Some are parasites also. Some are parasites like leech. Leech is parasite here. Level of organization is organ system. Please remember these. Level of organization is organ system. Symmetry is bilateral symmetry. Bilateral symmetry. Germ layers, according to germ layers, it is triploblastic. It is triploblastic. Coelom, they have proper coelom, true coelom called schizocoelom, or you can say just normally as coelom only. Segmentation definitely present. Metamerism present. Metamerism present. Are you getting my point? Digestion, complete digestive system. Here they have complete digestive system. Complete digestive system here. Circulation, they have very important system, that is closed blood circulatory system. They have closed blood circulatory system. Moreover, blood is dissolved, hemoglobin is dissolved. Hemoglobin is dissolved in plasma in earthworm. Hemoglobin is dissolved in plasma in earthworm. Please remember, very important point. The blood is red in color in earthworm. In some annelids it may be green or something, but in earthworm it is red. And the hemoglobin is not found in RBC, it is dissolved in plasma. Did you get it? Excretion by nephridia. There are special structures called nephridia by which excretion takes place. Reproduction is by sexual means. Okay, so reproduction again by sexual means here. Is it clear? Development is usually direct. Usually it is direct. Please write down here. Development is mainly direct. Yeah, mostly it is direct. That means larval stages are not present. Okay, but in some cases you can find some trochophore larvae. We will not discuss that much details. That is, you remember, it's mostly direct. Is it clear to you? Now next phylum you see is Arthropoda. So phylum Arthropoda we'll discuss now. First talk about the crustaceans here. So these crustaceans you see, these are characterized by, crustaceans are characterized by cephalothorax. So you can see cephalothorax here only, which is covered by a carapace in the prawn. Two pairs of antennae are present here. You can see two pairs of antennae. Two pairs of antennae are present in them. One pair of compound eye is present, and the excretory organ is green gland or antennal glands. They are also called as green glands. They are also called antennal glands because present in an antennae region. Got it? So this is Daphnia, that is water flea. Common name is water flea. Cancer is the crab, and Paleomon is the prawn. Common names, please remember that. Next, you see the class, this class is called as the centipedes. Centipedes are characterized by, formation, they are having head and trunk. They don't have head, thorax and abdomen. The thorax and abdomen are fused and form a trunk here. So what is trunk here is thorax plus abdomen. Please write down. Thorax plus abdomen is the trunk. So they have head and trunk in that region, one pair of leg on each trunk segment. You can see one pair of leg on each trunk segment, you can see. And the first pair is poisonous also. First pair of leg is poisonous claw. You can see this is a poisonous claw. One pair of antennae is present in them. So this is the Scolopendra, commonly called centipede. Next comes the Jullus. Jullus is the millipede, and millipedes are characterized by, these millipedes are characterized by head, thorax and abdomen. Moreover, they have one pair of legs on each thoracic segment. One pair of leg, it's one pair of leg, not one leg. It is one pair of leg on each thoracic segment, except the first one. Except the first one. First one doesn't has. Next you see two pairs of legs on each abdominal segment. So each abdominal segment will have two pairs of legs. Each abdominal segment will have two pairs of legs. Each thoracic segment has one pair of legs, except the first one, and one pair of antennae are present in them. So you see this is non-poisonous, this is poisonous. The Scolopendra, that is centipede, is poisonous because they have poisonous claw. Jullus is non-poisonous. Okay, let's continue further. The next class is your Insecta. Insecta is also called Hexapoda. Why it is called Hexapoda? You see, because they have 6 legs in them. So they have 3 pairs of legs. That means 6 legs are present in them. That's why they are called Hexapoda. Body is different. Your head, thorax and abdomen. They also have one pair of antennae, one pair of compound eye, and in some cases simple eye may also be present, which may be vestigial in some cases. Excretory organ is Malpighian tubule, and respiration by tracheae. Respiration by tracheae. One very important point to remember, that no respiratory pigment in insects. Very, very important point. There is no respiratory pigment in insects. Moreover, insects undergo metamorphosis also. Insects usually undergo metamorphosis. Not all, but they usually undergo metamorphosis. Most insects undergo metamorphosis. Clear? This is Locusta. Locusta. This is the Lepisma, that is commonly called silverfish. This Locusta is a gregarious pest. Please remember, gregarious pest. Cockroach. You know cockroach, Periplaneta americana, very commonly known. This is everyone knows. Next is your Arachnida. Next class is your Arachnida, in which you have the Pelamnius. Pelamnius is the scorpion here. So scorpion belongs to arachnids. Even the spider, that is Arania. Arania, that is a spider here, that also belongs to what? That also belongs to the Arachnida. They have cephalothorax. They always have 4 pairs of legs. Antennae is absent. Very important point is antennae is absent in them. And they have simple eyes, no compound eyes. Coxal gland is the excretory organ in them. Excretory organ is coxal gland. The book lungs or trachea may be present, or in some cases they may be both. So book lungs are characteristics of the arachnids. They are the respiratory structures of arachnids. Book lungs. Please remember, book lung in scorpion, book gill in limulus. So let's talk about book gills. So the respiratory organ in the limulus is book gills. Please remember this point, very important. Here you see, this is a living fossil. Living fossil. Limulus is a living fossil. Periplaneta is a connecting link. This is a connecting link between annelids and arthropods, because they have characters of both. So annelids and arthropod connecting link is the peripatus. Please remember, this is also called walking worm. This is also called as walking worm. That means worm which has some appendages to walk. Arthropoda is characterized by, you see some general characters on this. Habitat, you can find arthropods everywhere almost. So found everywhere, found in all habitats. Even you can find in water. Okay, in terrestrial also. Level of organization, you know, that is your organ system level of organization. Symmetry is again bilateral here, very important. Germ layers, it is triploblastic. According to germ layers, it is triploblastic. Coelom is definitely present. True coelom present. True coelom present. Next you see segmentation. Segmentation present. I told you there are three phylum. Annelids, Arthropoda and Chordata who has segmentation. Please remember. Digestion, it is complete type. They have mouth and anus. Circulation, it is open type. Please write down open type circulation. Very important here. Open type circulation. Excretion by coxal glands, Malpighian tubules, coxal glands. These are all involved in excretion. And excreted product is uric acid usually. Uric acid is excreted product. So they are uricotelic. According to that, they are uricotelic. Please remember that. Reproduction. So sexes are usually separate. Sexes are separate. Okay. Sexes are separate here. And the fertilization is internal. Fertilization is internal. Please write down fertilization is internal and development is usually maybe direct or indirect. Direct or indirect. Is it okay with you? It's very easy. Just remember those few points. Okay. So let's continue with the next phylum, that is Mollusca. Now molluscans are soft bodied animal. Soft bodied animal. This is the second largest phylum in terms of number of species. So the arthropods, this was the one which is the has maximum number of species. Maximum number of species I am talking about, not the number of organism. Please remember, I am talking about has maximum number of species. Almost 2/3 of all species on earth are arthropods. Okay. Second largest phylum is your Mollusca. Molluscans are characterized by shell. You can see here, there is a shell here over the body. There is a shell characterized by mantle. There is a visceral mass inside. You can see the visceral mass inside here, and there is a foot here. So you can see it is characterized by shell. Okay, visceral mass, even the mantle and the foot. The molluscans are very soft in nature. Their body is very soft, so they are covered by usually shell. The shell may be external or internal. So this is the major structures which have been shown. Inside you have a structure called radula. Radula is a part of digestive system. It's a part of digestive system. Part of digestive system. So radula is a part of digestive system. Okay. It helps to crush the food into small pieces there. Next you see the visceral mass is inside, that is a complete body here. Mantle is just over the visceral hump, you find the mantle, and between the mantle and the visceral hump you find a cavity called mantle cavity. So mantle cavity you can see here, and this is the gill called Cnidaria. This is called gill called Cnidaria. They are not proper gills, but they are Cnidaria here. Okay, let's see. So here you can see this is dentalium. Dentalium is called as elephant tusk shell. This is called elephant tusk shell. Can you see that? Captacula. Captacula in them are used for capture the food. Neopilina. Neopilina is the only segmented mollusk. Only segmented mollusk, which is very primitive. Okay. Pink Tata is a pearl oyster. This produces pearl. This is a pearl oyster which produces pearl. Okay. Is okay with you? Let's talk about the chitin. Chitin. Chetoplura. Chetoplura is commonly called as chitin here. You can see 8 plates are there. 8 external plates. So these are 8 external plates found on the body. Shell, that is shell here. Okay. This forms a shell here. Octopus is called as devilfish. This is the devilfish. Common name is devilfish. Cuttlefish is the sepia. Cuttlefish is the sepia. Phyla is your apple snail. Your common name you should please remember. These are given in NCERT also. Please remember it. Aplysia is the sea hare. So these all names please remember and just see the picture so that you can understand. You have a picture memory also. Please understand these things. Doris is sea lemon. Common name of Doris is sea lemon. A squid is loligo. Loligo is the squid. There is a difference between the squid and cuttlefish. You can see here. The body is narrow here. It is much more wider here. Can you see that? The study of molluscans is called what? What is the study of mollusca is called what? A study of mollusca is malacology. A study of molluscan shell. Study of mollusk shell is called conchology. So there are very interesting things in molluscans also. We'll understand all these things later. First you remember the general characters. General characters, if I talk about what is the habitat, mostly they are marine. Mostly they are marine. Level of organization is again organ system. Organ system level is the organization here. Symmetry, very important question comes from here. Symmetry is bilateral. Please remember, symmetry is bilateral. Important point. Please write important here. They ask this question. Germ layers are, according to germ layers, they are triploblastic. They are triploblastic. Coelom is present. True coelom present. Segmentation, very important again here. Some question comes in NEET here. No segmentation. Segmentation is absent. Segmentation is absent. Please remember, this is also important here. Some few points they will keep on asking. So better when they ask these questions, you should be well prepared for that. Digestion, complete. They have the mouth and anus. Circulation. Circulation is open type. Please write down again. Important here. Important. They ask these few questions. So please remember those. Excretion. Excretion, they are usually amonotelic. They are usually amonotelic. Amonotelic. And sometimes they are uric acid also. Uric acid also they secrete sometime. Some molluscans secrete uric acid also. So in that way they are uricotelic also and amonotelic also. And kidneys, they have kidneys. Kidneys present. Okay, and sometimes they call it, we call it as organ of Bojanus. Organ of Bojanus. Is it clear with you? Now you see reproduction. Sexes are usually separate here and they go for a sexual reproduction. Development is indirect. Development is usually indirect. It may be direct also, but usually it's what? Indirect. They have larval stages. Is it okay with you? Next phylum you will see, that is Echinodermata. Echinodermata are called spiny skinned animals. Spiny skinned animals. Let's see what you mean by spiny skinned animals, because they have spines over the body. Starfish is called as asterias. Starfish. Ophiura is the brittle star. Please remember all these things. Brittle star is Ophiura. Holothuria is sea cucumber. Common names are very important. Again I am telling you, common names will be important for you. Echinus is sea urchin. Sea urchin is your Echinus. Antedon is sea lily. Sea lily is your Antedon. These questions only will come and students will not be able to do it because they don't take attention on this. They don't think that these questions may be asked. Okay, so be very careful. Anything can be asked from here. General characters we'll see. They are exclusively marine. They are not found in anywhere else. They are found in marine waters. Exclusively marine. Okay, they are exclusively marine. Level of organization, as you know, it's organ system level. It's organ system level they have. Symmetry is again here bilateral in larvae. In larvae they have bilateral symmetry. Adult has radial symmetry. Please write down, adult has radial symmetry. Okay, very important question here. Germ layers, according to germ layers, are triploblastic. Triploblastic. according to coelom, coelom is like humans, that is enterocoelom, not humans, actually chordates. So just like chordates, the coelom is enterocoelom in this. Segmentation, no segmentation, it is absent. Please write down, very important point, it is absent. Digestion, complete digestive system, complete digestive system present. Excretion, no excretory organ, no specialized excretory organ. There is no excretory organ in echinoderms. They are ammonotelic. Ammonotelic. That means ammonia diffuses to the body surface. That is only happening, but there is no specialized excretory organ in echinoderms. Locomotion. Tube feet help in locomotion. So the water vascular system is present, which helps them in locomotion. Reproduction. Sexes are separate. But no sexual dimorphism. That means you can't differentiate between male and female. Okay. And development is what? Indirect. Development is indirect here. Please remember these few key points. Okay. Let's start with the next phylum now. Next phylum is your Hemichordata. Hemichordata, earlier they were thought to be chordates, but afterwards they were thought to have proved to have not the true notochord. So they were placed in non-chordates. So they are not chordates, actually they are non-chordates because they have false notochord. They don't have true notochord. The stomochord, that is a buccal diverticulum extension, was thought as notochord. So they have stomochord present, which is extension of buccal diverticulum. So this is a part of digestive system, not a notochord actually. So this was earlier thought as notochord, which was wrong. You see, the body is divided into proboscis and the trunk. You can see proboscis, collar and trunk. Proboscis, here there is a collar here, and the complete entire body is called as trunk here. This is balanoglossus, this is saccoglossus. Both are commonly called as acorn worm. Okay, so let's move further. See the general characteristics. Habitat, again they are exclusively marine here. Level of organization, again organ system level of organization. Symmetry, again it's bilateral here. Germ layers, according to germ layers they are triploblastic. Coelom is enterocoelom, please write down enterocoelom. Respiration by gills. Okay, digestion is complete, they have complete digestive system. Circulation is open type, very important, this question will come to you, this is important here, this question usually asked. Excretion by proboscis gland. So proboscis gland helps in excretion. And the reproduction, if you see, the reproduction is, sexes are separate here, and development is indirect. Okay, fertilization is external here. Please remember these important points. This is usually asked in exam, so this is again important. Is it okay? So let's continue with the phylum Chordata. So why it is called phylum Chordata? Because of the presence of 4 major characteristics. What are those 4 major characteristics? One is the notochord, which is mesodermal, solid like. Okay, next you see the dorsal hollow nerve cord. This is dorsal hollow nerve cord. So dorsal hollow nerve cord, in future it will form the brain, gives rise to brain and spinal cord, gives rise to brain and spinal cord. Please write down brain and spinal cord. But whereas if you see the notochord gives rise to what? Vertebral column. This gives rise to vertebral column here. Is it okay with you? And post-anal tail is also a characteristic feature of chordates. So let's talk more advanced characters of chordates and divisions. So the chordate has been divided into subphylum Urochordata. Why it is called Urochordata? Euro means tail. So that notochord is only present in tail. Notochord only present in tail of the larvae. Absent in adult. Absent in adult. So notochord is absent in adult here. And the body is covered by a cellulose like material called tunicine. Tunicine is cellulose like substance which covers the body of the urochordates is tunicine. That is why the Urochordata also called as tunicata. It is also called tunicata. Phylum tunicata, you can say it commonly. Okay, and they have retrogressive metamorphosis. That means larvae is more advanced. You can see here, larvae is more advanced than the adult. Larvae is more advanced than adult. Examples you can see, they have open circulation. Very important here also this question comes. Now you see, this is pyrosoma, which you can see is bioluminescent. It's a colonial form. It's a colony. So it's glowing in the dark. This shows bioluminescence. Herdmania. This is a perfect example of urochordate. And the Acidia. These are all examples of urochordates. Okay. Next you see the subphylum Cephalochordata. Cephalochordata, in this case the notochord persists throughout life. So notochord is going to persist throughout life in this, and it is present from head to tail. They are exclusively marine. You can see here, these are cephalochordates. Cephalochordates have closed circulatory system. This is the lancelet, commonly called as lancelet. It is also called branchiostoma. This is commonly called as lancelet, and they have closed circulatory system. Please write down this important point. Is it okay? So let's see the classification of vertebrata. Now comes the subphylum vertebrata, as we talked about. So subphylum vertebrata is characterized by they are grouped under some divisions. Those two divisions are Agnatha and Gnathostomata. Agnatha means no jaws. A means no. So no jaws, that is Agnatha. Gnathostomata, that means they have jaws. The stoma means mouth. The mouth bears jaw. Okay, they have jaws. Class Cyclostomata, they have only one living class. Only one living class they have, that is Cyclostomata. And the superclasses of the Gnathostomata division has been classified into two superclasses. One is Pisces, other is Tetrapoda. In Pisces you have two living class, one is Chondrichthyes, other is Osteichthyes. In Tetrapoda you have four classes, one is Amphibia, Reptilia, Aves and Mammalia. So these are four classes belonging to Tetrapoda. That means they have limbs. Two pairs of limbs are present in them. That's why it's called Tetrapoda. Reptilia, aves and mammals, they are called as amniotes. They are amniotes. Rest, amphibia and these Pisces, they are anamniotes. They don't have an amnion layer over them. Over the embryo. Embryo doesn't have amnion layer over it. Okay. So let's talk about the details of them. So class Cyclostomata. Cyclo means round. These are, stoma means mouth. So they have round mouth. You can see they have round mouth here. Okay. So they have round mouth. That's why they are called what? Cyclostomata. They have 6 to 15 pairs of gill slits. Please write down 6 to 15 pairs of gill slits are present. Scales and paired fins are absent. Please write down important point. Scales and paired fins both are absent in them. In this case both are absent. Next case you see, in this case they are mostly anadromous. So you can say petromyzon. Petromyzon is anadromous. What is anadromous? Means they will migrate from sea water to fresh water for spawning purpose. Migrate from sea water to fresh water for spawning. Please write down important point. For spawning. Have you written this? Now you see they have closed circulatory system. That is also important point. Closed circulatory system. Development is indirect here. Development is indirect. Ammoseed larvae is a larvae of Petromyzon. Ammoseed larvae. Okay, Petromyzon. Petromyzon larvae is ammoseed larvae. Is it okay with you? So this is two differences. You can see there's a Petromyzon here and this is a Myxin, which is hagfish. So this is Petromyzon, which is sanguivorous. This is sanguivorous. Sanguivorous means it sucks blood. It is feeding on the shark and sucks blood from that. Myxin is bottom dweller. Bottom dweller, and it is scavenger usually. Usually a scavenger. Is it okay with you? Commonly this is called as slime eel. Commonly this is called as slime eel. Okay. So let's move to the next class, that is chondrichthyes. Chondrichthyes commonly are called as cartilaginous fishes. So they are commonly called as cartilaginous fishes. Cartilaginous fishes are characterized by some characters like mouth will be always ventral in them. Can you see the mouth is ventral here? The mouth is always ventral. Okay. Moreover, they have placoid scales on the body. Scales are placoid on this. Air bladder is absent. Air bladder is absent in them. Fertilization is usually internal. Fertilization is internal in them. Okay. And moreover, if we talk about, they go for the sexual reproduction also. The males have claspers for that. Males have pelvic claspers. Please remember, like penis, they have pelvic claspers which help in copulation. Which help in copulation. Now let's see some common names. Scoliodon is called as dogfish. It is not called dogfish because the mouth looks like dog or it barks like dog. It is called dogfish because it has great sense of smell. So great sense of smell is the characteristic of this. That's why it's called dogfish. Pristis is commonly called sawfish. Pristis is commonly called as sawfish. And some few more chondrichthyes you see. There is torpedo. This is the electric ray. Electric ray. Trigon is stingray. Stingray. Carcharodon is a great white shark. Please write down important points. The common names also. Great white shark. Is it clear? So you understood about the general characters which we talked about. Few general characters we talked about, like mouth is ventral, placoid scales are present, air bladder is also absent in them. So fertilization will be internal in this case. Few more important points are there, like scroll valve is present in intestine. Scroll valve present in intestine. Okay. Moreover, their gills are not covered by operculum. So gills not covered by operculum. Cloaca is present in them. Cloaca present in them. Is it okay with you? Have you understood about the chondrichthyes now? Let's see the osteichthyes now. Osteichthyes are commonly called as bony fishes. These are commonly called as bony fishes. Now in bony fishes, common fishes you can see rohu, katla and the clarias. Clarias is also called as magur in Hindi. It's called magur. Okay. So what are the characteristic features of osteichthyes? Mouth is usually terminal or subterminal. Terminal or subterminal. Let's see about the other characters of osteichthyes. In this case, operculum present. Operculum is present and covers 4 pairs of gills here. 4 pairs of gills are covered by operculum. Air bladder is present. air bladder is present. Moreover, fertilization in them is external. Fertilization is external. Let's see some more, fishes here. This is the seahorse. Seahorse. This is a marine bony fish. This is a marine. Okay. Mostly the condycthes are always marine, but in bony fishes you have few marine bony fishes also. Beta is a fighting fish. Common name is fighting fish here, and pterophyllum is called as angel fish. Okay, so these two are your aquarium fishes. These are kept in aquarium, so these are aquarium fishes because of their beautiful looks. Okay, so this is the only fish which swims in this position, otherwise the fishes move horizontally. This moves vertically. Got it? So osteichthyes, some few points more you can write. They are amonotelic. Mostly they are what? Amonotelic. They have cycloid or ctenoid scales. The scales in them are cycloid or ctenoid. Clear to you? Let's move further with the next class of osteichthyes only, and we will talk about some lungfishes. So lungfishes belong to a order called Dipnoi. They belong to a order called Dipnoi. Okay, so the lungfishes, you can see three lungfishes have been shown to you. One is Neoceratodus, other is Lepidosiren, and next one is Protopterus. So Protopterus is your African lungfish. This is the African lungfish. Please remember only the common name. African lungfish. Lepidosiren is South American lungfish. This is South American lungfish. And Neoceratodus is Australian lungfish. Okay, so these were the common names only required, not required much about this. Next class we'll talk about is amphibia. Amphibia, as you know, the scales are absent in them. Scales are absent. Okay, this is Rana tigrina, that is common frog. Bufo is the toad here. Common names very important, please remember. Hyla is the tree frog. Now amphibians are characterized by some very important characters like two pairs of limbs. Start from here only. Two pairs of limbs. They are the first tetrapods. Okay, so they have two pairs of limbs in them, and the respiration can occur by skin, that is cutaneous respiration. Cutaneous respiration can happen in them. Even they have buccopharyngeal respiration. Buccopharyngeal respiration. Even they have pulmonary respiration in some cases. Now they are characterized by very important characters like the first time the sound producing organs developed in amphibians. Sound producing organs developed in them. Developed first time here. Is it okay? They have a cloaca present. So cloaca is present in them. Cloaca is when abdominal and urinary and reproductive structures, they all open into the common, they come out through the common opening, that is called cloaca. So cloaca is present in them. Moreover, if we talk about some more characters of the amphibians, so fertilization is usually external in them, and that requires some water medium. They don't have external ear, so they have tympanum for that. So tympanum represents the external ear. Please remember, tympanum is important. Tympanum is representing the external ear. Is it okay? Development is usually indirect. They have larval stage. For example, tadpole, for the rana. So you can say development is indirect here. Is it okay? Let's move to the next, some more members of amphibia, that is salamander. This is the salamandra, is a common salamander. The scientific name is salamandra. Ichthyophis is a limbless amphibian. This is limbless less amphibian. So a study of amphibians, if I talk about a study, is called as batrachology. Batrachology. Okay. They are mostly not found in marine environment. They are not found in the colder regions. So not found in colder regions. But except some salamanders. Salamanders are present. So except salamanders. Okay. Salamanders belong to a group called Urodela. So North America is called as headquarter of Urodela. North America is called headquarter of Urodela. Mostly development took place there only. So it's called headquarter of Urodela. Is it okay with you? Now let's talk about the next class, that is reptilia. So reptilia have cornified skin. Cornified skin. They are characterized by creeping movement. Creeping movement here. Okay, and these are common names you can see. The testudo is the tortoise. The testudo is the tortoise. Chiron is the turtle here. Garden lizard is your calotes. All scientific names you should remember. House lizard is Hemidactylus. Remember, it is Hemidactylus. Naja is the Indian cobra. Please remember all these scientific names and common names both. Chameleon is the tree frog. So please differentiate between the tree frog and garden lizard. Okay, tree lizard and the garden lizard. So tree frog is your hyla and tree lizard is your chameleon. Okay, so you can see you have to differentiate with the tree lizard, garden lizard. You have to differentiate. You can see here prehensile tail. Prehensile tail will be a characteristic of chameleon, that is tree lizard. So now reptiles are having two pairs of limbs in them. Okay, they have 12 pairs of cranial nerves. 12 pairs of cranial nerves in them. Study of reptiles is called as herpetology. Herpetology is a study of reptiles. If I say ichthyology, if I talk about the saurology, okay, saurology is a study of lizards. And if we talk about the ophiology is the study of snakes. Is it clear? So you see now differentiate between crocodile, alligator and gavialis. Can you differentiate between crocodile, alligator and gavialis? You see the color of this is bit olive green. Here you can see steel gray. So olive green color you can see here. This is olive green. This is steel gray. Okay, and you can see here this has a very pointed snout. So pointed snout is a characteristic of what? Pointed snout is a characteristic feature of gavialis. So this has been, deliberately it has been left blank, and that I want you to answer in the chat box what is the name of this organism and who gives the first answer. I'll keep checking that. Whosoever gives the first answer, I'll connect with them personally and let's give them a prize also. So it is not the same. You have to tell me what is the name of this organism. How is it different from the others? Okay, so let's continue with the birds now. The class aves. So study of aves is called as ornithology. Ornithology is a study of birds. They are characterized by some pneumatic bones. They are characterized by pneumatic bones. These are hollow bones. Pneumatic means the hollow bones. But their bones are fully ossified. Don't think that bones are not ossified. The bones are long bones are fully ossified. You can't say it's not ossified, it's fully ossified, okay, but it's hollow. Clear? Moreover, they have two pairs of appendages. So the four limbs are modified into wings. The four limbs modified into wings. Characteristic feature is the presence of feathers. Characteristic feature is presence of feathers. Characteristic feature is presence of feathers. Please remember this, very important. So you can see feathers are present in all. Correct? Let's see. The study is called ornithology, and Thomas Huxley called it as glorified reptiles. He called the birds as glorified reptiles. Is it okay with you? They are mostly oviparous. So they are what? Oviparous. Without exception, they are oviparous. Presence of beak is there in them. Beak is present, okay, without teeth in all modern birds. Beak does not have teeth. Beak is present without teeth in all birds. Is it okay? Let's see Neophron. Neophron is the vulture here. Pavo is the peacock. Corvus is the crow. Columba is the pigeon. Sitacula is the parrot. You can see also, you can remember also. You can, by seeing only you can tell. It's very easy for you. Now this is some more birds which are flightless birds if I talk about. So this is penguin. Penguin is Aptenodytes. Struthio is the ostrich. Struthio is ostrich. So urinary bladder is absent in birds. Urinary bladder is absent in birds. Absent. Except one or two it may have. They don't have any sense of smell. Sense of smell is very poor. Sense of smell very poor, except one bird, that is kiwi. Okay. So urinary bladder is also absent except rhea. Rhea is a ostrich, South American ostrich. Okay. So we were talking about the birds. So they have some air sacs which help in the supplementing the air. So air sac will supplement lungs. So air sacs will be supplementing what? Supplement lungs. So they have 9 air sacs. Each they have 9 air sac. You can write here 9 air sac are present. 9 air sacs are present in total. Is it okay with you? So let's continue further with the class mammals. Now mammals, as you are also mammal, they are characterized by presence of mammary glands. Characteristic feature is presence of mammary glands. Mammary glands. Moreover, they are characterized by presence of sudoriferous glands. Sudoriferous glands means they are also called as sweat glands, and they have sebaceous glands also. Sebaceous glands and sudoriferous glands are present in mammals. They are characterized by 7 cervical vertebrae. 7 cervical vertebrae is present. Let's talk about the common examples. Pteropus is your bat, or the flying fox also they call it. Flying fox. Ornithorhynchus is the duck-billed platypus. It is the only one which is oviparous. Duck-billed platypus. Okay, even some more are there. Species. Balænoptera is the blue whale. Delphinus is a dolphin. You can see all these and remember very nicely. Dolphin. Maca is a monkey here. And the few more examples you can see. Macropus. Macropus is the kangaroo. This is the kangaroo which you see. Panthera tigris is the tiger. This is you, Homo sapiens, and Camelus is the camel. Okay, so very important is that you should remember those common names, and some general characters also are very important to remember. For example, if you talk about the mammals, few characters I have told you, like 7 cervical vertebrae. So this is not in all, exceptions are there, except in some cases they may be 6 also, like except manatee and one sloth, two-toed sloth, they have not got 7 cervical vertebrae. There's an exception. So mammals are full of exceptions, but the only structure which is without exception is diaphragm. Mammals, all mammals without exception have diaphragm. So diaphragm is present in all mammals without exception. They are viviparous, yes, but then again you have the ornithorhynchus, you saw na, which is oviparous. So mostly they are what? Mostly viviparous, but few are oviparous also. So wherever you see there, you will find some exceptions, but in only one case you don't have any exception, that is diaphragm. Fertilization is internal always. Fertilization is always internal without exception here also. Okay, so fertilization is internal without exception. So this was all about the theory part. Now we'll be discussing the previous year questions and the multiple choice questions on these topics. So let's talk about, read the following statements. This question came in NEET 2021. Okay, so let's see what was the question. Metagenesis is observed in helminths. Sorry, it is observed in your cnidarians. So cnidarians will have metagenesis, that is presence of polyp and medusa. Echinoderms are triploblastic and coelomate animals. Absolutely correct. Roundworms have organ system level of body organization. That is also correct. So you go correct as B and C. Comb plates present in ctenophores help in digestion or they help in locomotion. So comb plates help in locomotion, not in digestion. Water vascular system is characteristic of echinoderms. Absolutely correct. Water canal system is present in Porifera, but water vascular system is present in the echinoderms. Is it clear with you? So let's continue. So choose the correct answer from the options given below. So you have to choose the correct answers. So B, C and E. You have to find out B, C, E. So I think first one only says B, C, E are correct. It's very easy for you. NEET 2021. So whatever you studied, you can do these questions very easily. That means you are done. Match the list 1 with list 2. This also came in NEET 2021. Metamerism is found in which phylum? I told you metamerism is found in annelids, arthropods and chordates. That you remember. So metamerism, annelids. You go for third here. Okay. So you go for A. Canal system. Canal system is present in Porifera, I told you. So you go for B here. Comb plates are present in Ctenophora. That you go for C here. Cnidoblasts are present in coelentrates. To go for D. So 4, 3, 2, 1. Wherever you find 4, 3, 2, 1, you can go for it. Okay. So it's metamerism is for, sorry, A, you have 3, then 4. 3, 4, 2, 1. 3, 4, 2, 1. You have to go for 3, 4, 2, 1. 3, 4, 2, 1 is the D option. So you can go for D option. You can just match and just see the answers. Okay. So NEET 2021, these easy questions came. So you can do it. Not a big thing. Bilaterally symmetrical and acoelomate animals. So again you see acoelomate, bilaterally symmetrical. Simple, without seeing the options also you can tell who is acoelomate. Only one phylum is acoelomate, that is Platyhelminthes, and he is saying bilaterally symmetrical. So you just find out Platyhelminthes in this, khatam, simple, easy. Okay, let's see the next question. Which of the following animals are true coelomates? true coelomates. See the question properly. With bilateral symmetry. So they are true coelomates with bilateral symmetry. You simply got the answer here first only. Okay, you don't have to, because adult echinoderms are radial symmetry. They have radial. Aschelminthes have bilateral, but they are pseudocoelomate. Pseudocoelomate. Okay. Platy are bilateral but acoelomate. So we can't go for these options. We have to go for the annelids. Easy. Let's see the NEET 2020 question. Gregarious pest, that is polyphagus and gregarious pest, is your locusta. Simple. So you go for A here. Adult with radial symmetry. Usually they are echinoderms which have adult as radial symmetry. So you find the echinoderms, that is the starfish. So starfish, you see, I was telling you that scientific name is important. If you don't know the scientific name of starfish, you can't do this question. So that is the why we are putting emphasis on that. Got it? Now, and larvae with bilateral symmetry, that is absolutely correct. That is your starfish. Book lungs are found in scorpion. So you go for here as C. Okay. And bioluminescence is present in Ctenophora. So you go for D. So let's see now. 4, 1, 2, 3. 4, 1, 2, 3 you find out. 4, 1, 2, 3 is your first option only. Simple questions are coming. They're not tough. So don't go for studying too much of animal kingdom. Whatever is there in NCERT, if you study properly, that's more than enough. 1 or 2 points extra you can study. Now, which of the following statements are true for the phylum Chordata? NEET 2020 question again. In Eurochordata, notochord extends from head to tail. So where you find in the Eurochordata head to tail? Wrong. In vertebrata, notochord is present during the embryonic period only. Yes, because that notochord is going to change into what? Vertebral column further. Central nervous system is dorsal and hollow. Yes, absolutely, because this is the one which is forming the brain and spinal cord. Chordata is divided into 3 subphylum. Hemichordata. Hemichordata is not a chordate. This is wrong. Okay, they have subphylum as Urochordata, Cephalochordata and Vertebrata. So this is absolutely wrong here. So you have the correct answer as third and you have the correct answer as 2. So 2 and 3 are correct. So wherever you find 2 and 3 you can go for it. Okay, easy. You saw that questions are again very easy. Match the following columns and select the correct option. Match this. 6 to 15 pairs of gill slits. So 6 to 15 pairs of gill slits are present in cyclostomes. I told you 6 to 15 pairs. Wherever you see, the keywords are coming from NCERT. 6 to 15 pairs, you go for cyclostome. Simple. Only the keywords are there. Heterocircle caudal fin. That means the tail fin will be like this. So this is a characteristic feature of Chondrichthyes. Heterocircle caudal fin. And homocircle caudal fin is a characteristic of, this is Osteichthyes. Osteichthyes have this type of heterocircle fin, a homocircle, and heterocircle is found in Chondrichthyes. Okay, so this is heterocircle and this is homocircle. So let's see. So this will be a characteristic of Chondrichthyes. You go for B here. Air bladder is found in Osteichthyes. You go for C here. Poison sting. I told you poison sting is found in the trigon. Trigon is called as stingray, with the dorsal fin is modified as poison sting. So you go for D here. So simple, you saw it's 2, 3, 4, 1. So wherever you find 2, 3, 4, 1, you can go for it. 2, 3, 4, 1 you are getting here. Is it clear with you? Again, you see the questions are very easy. Now again, NEET 2021 question we'll see. Now Phaselia common name is Portuguese manowar. Simple, you go for A here. Limulus, you see in 2021 they have just asked you the common names only. Common names. Nothing they have asked. Common names they have asked you. Limulus is living fossil. That is B. Ancylostoma is hookworm. You go for C. Pink Tata is the pearl oyster. You go for D. Simple, you saw. Which suppose this type of question means, that means they want you to understand the basics of animal kingdom. They don't want that you should go into too much of depth. Okay, so let's finalize this. 2 3 4 1. Again 2 3 4 1, you got it. 2 3 4 1, where you get D option. 2 3 4 1. So this is all what you studied today. You please apply that. Just remember those common names and few important characters. Thank you. All the best.